Okay, I think we will get started. Welcome to everyone today to the last ISBS webinar of the year. I am honored today to preside over a panel of distinguished scientists in academia and industry. And today we will be discussing the sort of the FDA draft guidance on master protocols that came out at the end of last year. I will put my slides. I am Peter Mesenbrink. I am from Novartis. I am the global group head for rheumatology and musculoskeletal diseases and biostatistics. I'm also the working group chair for the regulatory working group for ASA ASA Dashi. So let me go here. And actually, first, where is share between? There we go. There we go. So today we will be discussing platform trials shaping the regulatory guidance for future innovation. I have a very distinguished panel of 7 people from across industry and academia. And today, as I welcome you, first I will allow all of my panelists, if they so desire, to give opening remarks. Then we have put together a planned set of questions accompanied by response and discussion from the panelists. And then at the end, as time permits, we will take audience questions of the panel. So with that, I will allow for the panelists to all introduce themselves and give any opening remarks.
So, Peter, should I start then? Is that what you're?
That would be fine, Bob. Absolutely.
Just introduction, or should I go to my slides also?
You can do introduction and your opening remarks. Yes.
Okay. So I'm Bob Beckman. I'm a MD, but also with a interest in math lifelong, and I'm a professor of oncology and biostatistics, bioinformatics, and biomathematics at Georgetown, and I advise the senior VP for research there. And actually, I should have clarified on the title that I'm going to give a brief interdisciplinary perspective on non-concurrent controls. So next slide, please. So non-concurrent controls have a number of important benefits that they might be able to deliver by reducing development time and cost. There's earlier access to life-altering drugs, and depending on political outcomes, if the cost of drug development is reduced, then the cost of drugs might be reduced for patients. And of course, it's a huge public health problem that, at least in the United States, that many patients can't pay for their drugs, or they have to choose between their drugs and their rent. And also, this would provide more cost-effective development options for sponsors and allow more trials to be funded, and especially in small markets like rare diseases and small biomarker-defined subsets of larger diseases. There might be more development. We certainly have a few dozen examples of beautiful approvals in rare diseases, but there are 7,000 to 8,000 rare diseases affecting tens of millions of people, and many are severely debilitating and life-threatening conditions of children. For trial participants, there would be less exposure to placebo or other control therapy. Rarely does a patient enroll in an experimental study in the hope of getting control therapy. Next slide, please. On the other hand, there are risks of non-concurrent controls, and insofar as they may be somewhat less rigorous than concurrent controls, we want to make sure that the confidence that patients have when there's an FDA or EMA approval in a drug, that the drugs are truly safe and effective. We want to make sure that confidence is maintained and not reduced. And similarly for trial participants, they would like to believe that they're contributing to science and to a scientifically valid conclusion. So we have sources of bias, such as stage migration, where improved imaging techniques lead to people who are led to people being categorized differently over time. And if you compare different times, you will probably give someone a more advanced stage if you have more advanced imaging. Changes in supportive care also may affect things over time. And of course, these things can be modeled, but modeling is imprecise, not always correct. And the risk of bias can be reduced, but not eliminated. Next slide, please. So thinking about this, this is how I view it. Protecting against bias is most important in phase 3. Non-concurrent controls are most valuable in severe conditions with no credible standard of care, and in rare conditions, and certainly even in phase 3. And when we decide whether we want to do non-concurrent controls, I think we ought to really consider a decision analysis where the goal is maximizing public health. And so in some cases, we may want to use non-concurrent controls in phase 3 if we can show that the ultimate benefit to public health is worth the risk. And I mentioned on the slides a chapter on quantitative decision analysis, and also a qualitative decision tree for historical controls that was published by the DASU Innovative Design Scientific Working Group. And that's those are my remarks, Peter. Thank you.
Thank you, Bob. I am going to go back to the screen. There we go. Franz.
Good morning and afternoon. So first, many thanks to Peter for having organized this wonderful panel discussion on platform trials and how to shape the regulatory landscape for future innovation. I have had the pleasure to work on platform trials in a large project called EU-PEARL, which was funded by the European Union's Innovative Medicines Initiative. And importantly, it involved industry, academia, and regulators, and it involved discussion on both sides of the Atlantic. And so let me start with, why have master protocols and platform trials been introduced? Simply to make trials and drug development more efficient. And this comes mainly by sharing a common infrastructure and, for example, by sharing a common control group. However, the concept of master protocols and platform trials helped to rethink about the terms and concepts we have been taken for granted for many years, decades, sometimes even, you might say, coming from the last millennium. And during the last years, my thinking in some areas really have changed dramatically. And most likely we will discuss some of the topics in more detail later on. For example, some years ago, I would have always said the control of the study device error rate is key, like pointed out in the E9 guideline or in the European multiplicity guideline. Also, why would you think of using external or historical control? Back then, I would have said, why even thinking about anything else if you have a proper randomized study? And the primary analysis should be solely based on the randomized comparison and not be messed up by anything else, but maybe at a higher significance level. However, when new concepts are introduced, there's always a risk that there's a certain hype and people hear some terms like response adaptive randomization, Bayesian adaptation, because they think everything becomes faster, cheaper, and all problems are solved out of a sudden, it's really difficult to manage the sometimes wrong expectation. And you have to carefully think what are the key design and analysis elements and what are the assumptions behind it. And this is not always fairly communicated. And how can we guarantee that we generate data which are fit for regulatory decision making? And here, I have to really say that the FDA guidance, as the FDA has to be complemented for the draft guidance. As said, the EU Pearl project was more or less running in parallel when the guidance was written. And we also had some discussion with the FDA, like in webinars like today or in meetings discussing some platform protocols. And overall, I must say that the guidance is very balanced. And I don't think that there are any major issues I would disagree. So I would say it's really a good guidance document. And what we also learned that early discussions with regulators are important to agree on general concepts and generic tools. And when going back to some older regulatory guidance documents, we always have to keep in mind that the terms used in the older guidelines might not be one-to-one transferable in platform trials. We will also discuss it today, like the control of the study-wise error rates or how to use historical control data. And I have to say, for both these issues, the FDA guidance is quite open-minded. And at the end of the day, it's always the discussion about the variance bias trade-off. And ask yourself the question, do you only want to have trials with unbiased estimates? Or is the mean squared error what really matters? And platform trials reveal that there are much more interesting operating characteristics than the type 1 error rate, family-wise error rate, or power. So let's start the discussion on them. So I had to hand back to Peter.
Thank you, Franz. Over to Kert.
Peter, I just wanted to point out that if there were slides, we didn't see them.
I'm taking, unless people have slides, the only person who presented opening slides was yourself. That's why I wanted to be able to see the people as we do opening remarks. So the slides will come back up when the questions
Okay.
discuss. Over to Kert.
Hi, so I'm Kert Viele. I'm the Director of Research at Berry Consultants. We've been involved in dozens, 50 to 100 platform trials over the course of the last 2 decades. I also would like to, what Franz said about complimenting the FDA, about the draft guidance, complimenting Peter for organizing this. I'm going to keep my powder dry, so to speak, on the individual topics, but just as we go forward, I see the framework here of the guidance and this discussion as first, what extra assumptions come up when we're doing platform trials? So when we do this, what do we need to worry about that we didn't have to worry about previously? A second issue related to that is how do we assess those assumptions? So both within a trial, doing sensitivity analysis or whatever we need to do, and also across trials. As more and more of these platform trials are run, we're developing a database of completed trials where we can ask, are certain concerns, do they actually appear in practice or not? And then the third element that I hope we bring out is also how can we design in order to mitigate those risks? So if we're doing something like non-concurrent controls, we don't have to design to try to minimize the use of concurrent controls or to minimize resources. We can design keeping in mind that we want to also be robust to certain assumptions and both be robust and gain efficiency in platform trials. So are there clever ways to design to deal with the realities of the assumptions that a platform trial might bring to bear? So I look forward to this discussion, and thank you.
Thank you, Kert. Over to Karen.
Great, thank you so much for the invitation, and just to echo special thanks to Peter for getting this group together and for organizing us and keeping us moving forward. So I'm Karen Price, I'm Vice President at Eli Lilly and Company, and I've had the privilege of working on several master protocols, both externally, so master protocols where Lilly was considering putting a molecule into that protocol, as well as internally, where we have a number of single sponsor master protocols where Lilly is maybe evaluating multiple indications and or multiple molecules that would come through, in that case, a single sponsor master protocol. And just to be brief, I, in the beginning here, just wanted to take a moment to acknowledge how far we've come and kind of briefly reflect on some of the reasons why we've come so far with master protocols. Acknowledging that initially master protocols really started in oncology. And then now, if we think about master protocols today, they're in so many different areas, rare diseases, pediatrics, even common diseases. COVID antibody program played a major role, atopic dermatitis, Alzheimer's disease, you name it. And I think this is due to a number of reasons. I'll highlight 3 of them. I think first, there's obviously been a number of people who have really advocated for these designs and pushed the science over the years. And along with that, a great partnership between academia, industry, and regulatory. And so I think this type of a forum is just reflecting on that and continuing that partnership between these important groups. Also wanted to acknowledge the FDA's openness and willingness to advance innovation. Again, just as others have mentioned with this draft guidance, but also other forums such as the CID pilot program, which is now the paired meeting program. Lilly was a part of that a few years ago, and we submitted a master protocol in chronic pain. We gained a lot of experience through that. That protocol is continuing today. We've seen a lot of value from that. But we also saw a lot of, and we gained a lot from the interactions that we had with the FDA, and since then have launched a number of master protocols at Lilly. And then finally, I think the value that these protocols bring is really felt and seen by those who are involved, where we can see operational efficiencies as well as improving our scientific insights. So while there are some unique challenges, and as we'll go through those today, these are definitely outweighed by the benefits. But I think this discussion today will be important to understand any remaining challenges and how can we overcome those to really take master protocols to the next level. So I'm excited and hopefully we'll progress the ball a little bit further down the field.
Thank you for your wonderful words, Karen. I agree with everything you said 100%. Next up is Skip Nelson from J&J.
Yeah, thanks, Peter. So I'm Skip Nelson. I'm Executive Director of Pediatric Drug Development at Johnson & Johnson. I'm just going to make a couple of comments about the use of platform trials in pediatrics, and in particular about the opportunity that exists given that the pediatric plans are required for any adult development program absent a waiver. What that means, given that these plans are negotiated with the FDA and the EMA, that likely, although sometimes these plans are not fully transparent across companies, that likely we have similar designs and multiple companies are then required to do very similar trials. And that would, in my mind, create an opportunity to think about whether there's opportunities in that space to think about the design of platform trials to address these pediatric requirements. Now, there are some differences about pediatrics versus adult development, although I think the design issues, even though some of the issues may be different, the design issues are addressed in much the same way. But what is, in fact, I think, different here is that there's always going to be prior adult data in a setting where you're doing pediatric development following in conjunction with an adult development strategy. So therefore, there's always some adult priors. And so the issue of Bayesian statistics, I personally think, are best suited in pediatric development that is following or in conjunction with an adult development program. Now, that may give you some advantages inferentially and the like, but I think part of the main challenge in my mind is the operational infrastructure that's required and finding the space, if you will, that multiple companies can come together and to design and then execute pediatric platform protocols in a way that respects intellectual property, respects confidentiality. So I personally think that a large part of the challenges in the increased use of pediatric platform trials falls more in the operational side than in the design, statistical, inferential side, although those aspects are, of course, important. So and then just one plug, ICH E11A extrapolation. I mean, I think that's a document that people could look at and get some sense of the different options that are available for the design of a pediatric trial that leverages prior data that may be available from adult programs. So thanks, Peter. I'll stop there. Peter, if you're talking, you're on mute.
Thank you, Skip. I will, for your beautiful words, I will next go to Alex Smerdlov.
Yes, hi everyone. My name is Alex Smerdlov. I'm a senior director of biostatistics at Novartis. And thank you very much for inviting me. Thanks to Peter for organizing this nice webinar. So I'm supporting several disease areas at Novartis, including neuroscience, ophthalmology, and rare diseases. And I'm increasingly involved in methodological research on adaptive designs in clinical trials, most recently on adaptive platform trials. So we'll talk a lot about these designs. They hold numerous promises and at the same time bring additional challenges. I'm also co-leading a randomization working group, which is a cross-industry effort to investigate the merits of randomization in clinical trials. And so we know that randomization is an essential component of any clinical trial, especially those that involve treatment comparison, due to its potential to mitigate various experimental biases and validating the use of statistical tests. But in platform trials, randomization considerations are particularly important given the additional complexities that these designs bring to us. And so I'm really looking forward to discussing both methodological and operational challenges that are pertinent to the randomization element in platform trials. Thank you.
Thank you, Alex. Last but definitely not least, my esteemed colleague, Olivier Collignon from GSK.
Olivier, you're muted.
Oh, thank you. Sorry, I didn't see it. I just received the pop-up from Teams. So yes, I was just thanking Peter, obviously, for having invited me. So thank you, Peter, and saying hi to the audience. I'm not going to try to delay much the discussions. I think we're all really much looking forward to going into that. I think the only thing I would say is that I'm very happy to be here today because I moved to the vaccines groups a year ago now in GSK, and I had paused a bit my work on platform trials. So it's a nice way just before the end of the year to go back at it. So that was really, I was very happy to see that. So that's one thing. The second thing really I wanted to echo is that being myself a former regulator, when I spent a few years at EMA, I was really pleased to see that FDA guideline. I think there were loads of great wins in there, particularly around multiple testing. We'll have the opportunity to talk about that as one of the hottest topics on the matter. I thought that was an excellent guideline across the board, but that was really a key win for me. And really just a confirmation that regulators, they're not here to prevent innovation. They support innovation as much as we do. Like us, in private companies, they're here to provide the best drugs and medicines to patients, and the only thing they want is not to be shortchanged, right? In the data package we send to them. So again, great guideline. There's a few principal components about it that we're going to discuss today, and I'm very much looking forward to that. So thank you for having me.
Thank you, Olivier, and thank you to all. And so we'll now go through some of the pre-planned discussion questions that we put together. And we go to the screen and switch. Okay. So the first question we want to talk about a little bit is the blinding of treatment assignment. There's been much published on the complexity of blinding a treatment assignment across platform trials and in sub-studies. Are we losing context here And can best practice be generalized so they are applicable to all platform trials? Who would like to talk about this?
So, Peter, this is Skip. I guess let me just start with a conversation that I had a few weeks ago with Stefan Gold, who I assume some people know that have worked on the EU Pearl, and they're in the process of designing an adult-oriented trial platform to do a platform trial in treatment-resistant depression, I believe. And one of the complexities there is the different modalities that might exist, let's say oral versus IV versus sub-Q versus device. So I believe what they've done is they've established domains within those different modalities to where blinding would be simpler than it might be if you were crossing modalities. So that if you're combining, let's say, a sub-Q versus device, that might be much more complex in terms of the blinding that you would need to do across modalities. So I believe that that's, and I don't know if there's anyone else that would be familiar with that, but that's my understanding of how they're trying to approach the complexity of the blinding of treatment assignment by keeping it within domains, so that there wouldn't be one platform that had everything in it, but there would be platforms internal to each of these domains. I don't know if that helps kind of kickstart the conversation, but just thought I'd mention.
I think that is reasonable, and I think it's sort of the challenge of, okay, this will flow into the next topic of randomization too, as well as we have different open entries of different sort of modalities. And yes, there's a way of sort of splitting up by domain and sort of having sort of a factorial type design related to MOAs too as well. But does anyone else want to add in the group want to add anything?
Maybe I can add, because I'm also involved in this depression trial, which has just been mentioned. So yes, so I think we all agree that blinding is one of the key design elements besides randomization. However, when you carefully think about it, full blinding is by definition not possible in a perpetual platform trial, as you have to inform the patient which arm are currently open. So we never have full blinding anyhow in a platform trial. And full blinding across the whole platform trial on all ongoing arms might not be feasible or possible, because as just mentioned, if different mode of applications are possible or needed, oral versus EA, is it really worth having full blinding with multiple dummies? It might even become more and more tricky when you have to add new drugs later on, which might have a new form or new color of the pill. So do we want to introduce more and more dummies? I think no. So concept like two-stage randomization, first randomization to a sub-study and then to verum or matched placebos are really valuable strategy, so that at least on the sub-study level, blinding is guaranteed. I think this is the key issue. We have to look at least on a sub-study level, we have blinding guaranteed. However, the main question, which was already addressed before, it becomes whether you can use all concurrent control data for a specific treatment control comparison of interest, or just the one from the sub-study or from a subset of sub-studies. I think this is the key question. And I'm afraid, Peter, coming back to your question, this can only be discussed on a case-by-case basis. So I think the best practices have to evolve over the years and cannot be directly enforced or implemented. There's also no blinding possible when you want to utilize non-concurrent control data, because you know when a treatment arm is ended. So both randomization and blinding were implemented. It's neither a true randomization nor a true blinding, actually. It's a different form of blinding. It depends really on the indications, on the endpoint, on the application. And I said in the depression trial, which comes out of a EU Pearl, and the name is Pearl Diver, will start next year. I think approximately in September, the first patients should be enrolled. And not full blinding will be implemented, but as said, in the several domains. But the drawback is that we cannot share all control data, even not all concurrent control data. So we have different control cohorts. That's what you have to go for if you decide to do it within certain domains.
And I guess, Franz, the ongoing discussion that will happen on a case-to-case basis is where is the bar set if something's going to be used for the registration of new therapies. And obviously things are not going to be perfect and pure as we all hope they would be, but what is good enough to sort of be able to minimize sort of the bias and maintain the integrity of the data going between platform trials. They have many sub-studies, but excellent transition as well to the next topic. Does anyone else want to add anything?
Peter, this is Karen. I might just add a couple quick comments. So I agree with everything that's been said. And I think as part of it, it's always important to balance between the goal of using that information along with the impact to the patient, where if we're trying to blind across a number of different molecules that may be coming in that have different modes or mechanisms, delivery routes, that can be very impactful to the patient. So we need to make sure that we're balancing that. And then perhaps as part of as we're thinking about best practices cases where the endpoint is maybe more objective could play a role in if we're looking to utilize control information or other information where there is not blinding across the two molecules, the more objective the measure and things of that nature could come in to helping us think through where that line is. So just a thought.
No, completely true. And again, it may be hard in some situations. I know we want to have sort of general guidance, but I think that's where sort of the engagement with the regulatory authorities is important, because in a lot of situations, we want a one size fits all, but there really isn't. So we have to be able to understand contextually what it all means. And then that's actually a great transition to the next question. Good. Randomization is obviously been a big topic in the work groups I've been into in the platform trial space, particularly EU Pearl, as well as stuff with IDMC DASHU2 as well, is randomization ratios and adaptive randomization platform trials obviously have been a hot topic. I guess the question becomes, are we making it too complicated? And what are the best conditions of platform trial for adaptive randomization to be used? And what are some of the challenges that will be faced by teams if adaptive randomization is used? And are these challenges preventable? I know Alex is very passionate about this topic, so I will let him start.
Yeah, thank you. Thank you, Peter. This is an excellent question and great topic for discussion. So as we all know, randomization is an essential feature of any randomized clinical trial. And usually 3 merits are highlighted, or methodological strengths of randomization. So first, it mitigates the risk of experimental bias, most importantly, selection bias. Second, it ensures comparability of treatment groups with respect to known and unknown confounders of the outcome. And third, it validates the use of statistical tests in the analysis of clinical trials. And importantly, it can form the basis for statistical inference through randomization tests. So in regulatory literature, randomization component is actually covered very sparsely. So our working group, randomization working group, has conducted a systematic review of regulatory guidance on randomization, and it turned out to be relatively sparse. So it's increasingly getting more attention, but the general guidance is not very specific. Apart from that, randomization is a well-established and important element. Now, what we found in the systematic review that the methods of randomization that are covered in the regulatory guidance refer to the classic 2-arm, 1-to-1 randomized controlled, placebo-controlled clinical trial. And more recent designs actually go beyond that. So in platform trials, we no longer have those classic textbook conditions of a randomized clinical trial. So the considerations of randomization are particularly important in this context due to additional complexities and challenges that we're facing in the platform trial settings. So I would say there are two groups of challenges. The first one is the methodological or statistical challenges, and the second is the operational challenges. So the methodological challenges refer to just the nature of the platform trial. So the number of treatment arms that may be actually unknown upfront due to the perpetual nature of a platform trial. So it requires frequent changes in allocation ratios due to adding or dropping of treatment arms at interim. And in fact, allocation ratio is frequently unequal and may involve irrational valued proportions such as square root allocation, which is actually mentioned by the FDA guidance. And therefore, there are requirements for randomization procedure in a platform trial that it should actually be such that it maintains close adherence to the target allocation ratio throughout the trial. And ideally, it should have or it should possess the so-called allocation ratio preserving property, which means that the unconditional randomization probabilities are equal to the target values of the allocation proportions. The second challenge relates to the eligibility criteria that may be different across treatment arms. So for instance, some subgroups may be open to certain treatments, but not the other treatments. And then also, if we think about multi-center trials, and most of the platform trials actually are multi-center, and if we think about the intervention-specific amendments that introduce new treatment arms, so in practice, not all centers need to implement the particular intervention-specific amendments, and some treatment arms may not be open for randomization at the given center. So that has to be taken into account when designing a platform trial. And then the third challenge, which is equally important, I think, to everything else that I mentioned, is that there's a need to ensure balanced covariate profiles across treatment groups. And given just the nature of platform trials, given the multi-arm and potentially unequal allocation randomization trials, so that really requires careful considerations. Then operational challenges, of course, they have to do with the interactive response technology that is used to design this trial. So how do we really go about designing IRT, taking into account the frequent adaptations that are pertinent to platform trials, the opening and closing of treatment arms, adjustment of allocation ratios, and potentially different eligibility of the subpopulation, the blinding issues, and so on. So these are all great questions, and many of them do not have answers. So it actually creates an opportunity for us to work on methodological research. And then last but not least, I should mention that many platform trials use response adaptive randomization, where the treatment randomization probabilities are adjusted adaptively according to accumulating outcome data in the trial. And we know that the response adaptive randomization is a controversial topic, but actually it's getting increasingly more attention in platform trials. And just a quick snapshot of platform trial protocols in the COVID era shows that there are many trials that used response adaptive randomization. Okay, stop here.
So, Alex, I just wanted to ask a question, because we did some preliminary research into randomization with multiple biomarkers, and at least our initial investigation suggested that it might be important that those biomarkers are relatively independent of each other. And if they're being investigated in separate studies, if there is a significant statistical interaction between those biomarker states, that could pose additional challenges. And we didn't have time or resources to look into that further, but I'm wondering if you have looked into that issue.
Yeah, that's a great question. Thank you, Bob. I think what you're referring to is a additional complexity where randomization actually has to account for biomarker subgroups. And in fact, that, well, there are several types of biomarkers. Some of them are prognostic, others are predictive, which account for the treatment by biomarker interaction. And some of these randomization methods actually do that. There are examples of platform trials with randomization, response adaptive randomization implemented or tailored to the biomarker subgroups. So I'm happy to share some references, but it's by no means an easy question. I think it's quite challenging, actually. And also, I think we don't have a unified theory how to approach it, a universal solution. So far, what I have seen is it on a case-by-case basis.
Great. Kurt had his hand up. Kurt.
So I'm actually gonna argue against Brand here. I agree with everything that Alex said. I think that, but the initial question says, are we making things too complicated? Mm-hmm. And I would argue, if you know what you're doing and you have experience and you can run interims and all of the stuff that goes along with this, I'm fine with adaptive randomization. Obviously, I've written a lot about it. If I were trying to get into the platform trial space, if I were doing my first platform trial or my first couple, I think you can get almost or as good operating characteristics by doing something much simpler, like an arm dropping design, and would recommend that people do that unless there's a compelling reason to do the more complex designs. I think there's just, if you heard the discussion that Alex just gave and you're like, oh, platform trials are really complicated, that's platform trials with this extra bell and whistle. And if you have Alex, you should absolutely do it. If you're not Alex yet, or you don't have an Alex, there are other ways to do it. I'll add another research topic that I hope somebody takes on. A lot of the response adaptive randomization literature, it's in shared control designs where doing RAR often has the effect of increasing allocation to some arms and decreasing allocation to others. In platforms, RAR really has the effect, if it's a perpetual platform, it has the effect of slowing down exploration of arms rather than necessarily changing the sample size. And I'd just be curious if somebody takes on that problem and the effects. Thank you.
Absolutely, Kert. I would agree with everything you have said, because I think sometimes you get some impression, people say platform trial and the first response is response adaptive randomization. And I think because the expectations might be wrong. Yes, for some more promising treatment, it will be faster and more patients will be allocated. But as you say, what does it mean actually for treatments which have no effect or a moderate effect? I mean, then I know some people might say, yes, go for interim analysis with fertility, but this only will help for treatments which have no effect at all. And still you will have some moderate, which will be in the platform trial for much too long time. And another issue, which is sometimes not overlooked, not by the ones who are experienced, is safety. Because you have to carefully think, what does your randomization procedure actually mean for your safety assessment? In traditional trials, most DMC members are used to simply look at the absolute frequencies of severe AEs. If you talk about response adaptive randomization, since the time a patient is at risk are completely different, you have time trends, everything. So we really have to carefully think about how to structure more elaborated safety reports and also to train the people how it relates to randomization. I think this is not quite frequently done. And to facilitate response, so methods like response adaptive randomization, you have to have a short-term endpoint. And it's sometimes overlooked. What does it mean for the other endpoints? How does it relate to the effect sizes and correlation? And also the randomization tools we usually use are not fit for purpose to address the changes in allocation ratio. Also, not all the centers might have the same set of drugs and can implement the changes as quickly as some other centers. So I think there are a lot of operational hurdles connected, which are sometimes overlooked in the statistical applications.
Yeah, I was going to mention about the short-term endpoints, and I just want to amplify that because short-term endpoints are very tempting, but it does, usually the correlation between the short-term endpoint and the clinical benefit is unknown. Often.
So is there a value then in sort of building in sort of the evaluation of clinical utility across the platform trials so that we can sort of be able to look at, particularly in phase 2, be able to look at that sort of that and if you're trying to make a decision on what compound to advance, you obviously do need to take into account both parts. So should we be looking at that benefit risk in some sort of structured way through some sort of utility function to be able to make the best decisions? Any thoughts from the panel?
Yes, I think it depends on the objective of the trial. If the objective is to find one drug as quickly as possible above a certain threshold, then response adaptive is the one to go for.
Yeah.
If you want to find all treatments above a certain threshold and time doesn't matter, then MUM's design might be more appropriate. But I think we never hardly we look in the combination of what is the objective and how it relates to the randomization procedure.
Okay, that sounds good. So let's move on. Let's move on to the next topic. Combination therapy. This was in the feedback that came in from the registry. This was sort of a hot topic. Historically, in platform trials from the oncology, most trials were designed to evaluate new vascular treatments being added to some backbone treatment to determine if efficacy was additive or synergistic. How much regularly needs to be adapted to address this for future use in areas outside of oncology, where there is some use of combination therapy, particularly obviously in infectious disease, as well as in sort of the metabolic disease space. And I guess along with that is, what is the low-hanging fruit here that everyone should be aware of when designing such a study? Anyone want to give this one a go?
This is Kert. I'm actually gonna make the question what I wanted it to be.
Okay.
Which is, I certainly think combination therapies are important. I actually would argue in most clinical settings, we may be investigating one dimension of a treatment, but patients are treated in lots of different ways. There are lots of choices. If I'm doing something on a ventilator, I may be interested in what their positioning is, but I'm making all kinds of other choices about, high flow and various other things. I think factorial experiments in general are a huge opportunity for statistics, not just in platform trials, and that platform trials are going to need to include this. Platform trials, one of the biggest issues when you do a factorial is, what do you do with interactions? Do you include them? Do you not? How does that compare to how a non-platform trial would do it? You don't worry about the interactions. You don't even get to see them in a non-factorial. If an aspect of the factorial, if one axis of the factorial, one factor, the answer becomes clear. Do you publish what data? I'm not going to have answers, but I think that it's too obvious an opportunity for us to pass up. And I hope we advance this rapidly.
No, I mean, I think that makes, and I guess probably the best example out there that we can learn from is I guess the remap cap design from COVID, where basically what you're trying to look at sort of how we can combine different MOAs to be able to synergize, sort of optimize sort of the treatment response. I mean, and I think it's an area that I think would warrant sort of be a sort of the, I guess, not to make a pitch for the complex innovative design initiative at the FDA, but I think it's really the setting where we need more examples in the public domain that can sort of. I think people see this. Obviously, we all learned sort of factorial designs as we were trained as statisticians. But obviously, some people may, in this sort of setting, there may be a little bit of sort of a phobia, I guess, to be able to sort of design and implement something. So we have the opportunities to be able to work together and come up with good use cases and be able to build libraries of these types of designs. I think that will obviously definitely increase their use. Anyone else want to give this one a try?
Peter, let me just add maybe a complexity from a pediatric perspective that occurred to me as I looked at the question. One of the challenges in pediatrics, again, thinking of development in the context of adult development, is that the pediatric program, we'd like to start earlier, but often it's extended past the point of the adult approval. And so you may have a significant amount of off-label use that's going on, not only of the product that you're studying, but potentially other products that are not approved. And so you end up with being in two difficult situations, again, from an operational regulatory perspective, where the agencies may not want to have something labeled to be in addition to, unless what is being added to is in fact approved. And you might then, you're cutting your pediatric population significantly because the drugs that they may be on could be, quote, standard of care, but not approved. And then the other problem is that if in fact those drugs that are part of standard of care work, even though they haven't been fully studied and not approved for that, the chance of then separating your product from that standard of care because you've added it to that standard of care becomes highly complex. So I just point out that, and these populations are small, so doing a factorial is not going to happen in a pediatric trial. So I just point out some of the complexities of trying to tackle this in a non-oncology space.
So I'm going to answer just one quick. I do think that when you say a factorial experiment won't happen in a pediatric space, certainly with an interaction, an additive structure, factorial are often extra dimensions are free. So there's an assumption, but I'm not sure it's a worse assumption than separate trials on the two issues would arise. But your point on interactions are impossible. Yes, completely.
Yeah, right. Thanks.
But I do think, though, in areas where there are chronic diseases where there are not a lot of effective treatments, I think that are being studied by everyone who is, particularly in the immunology space, the immunology metabolic disease space, where I think there are opportunities to sort of be able to look at these concepts. I mean, one of the big things that's ongoing now through another consortia called Mosaic is we understand that sort of that in the metabolic disease space, people have generally multiple problems. So the concept of doing, say, a cardiovascular outcome trial sort of standalone may not be the way of the future, where obviously people may have other sort of risks and sort of ways to achieve outcomes. And in the era of the evolution of GLP-1s, where pretty soon I think everyone's going to be on a GLP-1 potentially, that there may be opportunities for certain things to say, okay, this is your backbone. What happens when you combine this mechanism, which has certain types of effects, with other drugs that you may know have a effect in it as well? And what is the effect synergistic? Do they interact with each other and sort of cancel each other out? How it's all about understanding the phenotypes of the patients too as well. I think if we do that, we can. I think these trials can have the potential in a lot of different areas. Go ahead, Olivier.
Thank you. Yeah, just the point I wanted to add is that the irony is that the point Robert made is that practically it would be very difficult to run a factorial design in pediatrics because of the size of the population. But at the same time, you want to use this type of design and making assumptions so that you gain power and reduce the sample size, etc. Right? And your question, Peter, pertained to basically to the regulatory acceptability of doing something fancy when testing combinations. And this is where you maximize the uptake, because it's much harder to come back to regulator and say, can I have a waiver on type 1 error control if my assumptions are wrong, when you're setting where you have plenty of available therapies. This is not so much an unmet need as compared to we clearly need something, right? So sometimes it's in a setting where it's harder to do it. Actually, you will have more chances to convince regulators to do it, because it's all about showing the assumptions you're making and then having the discussion with them. I think there will be more willingness for us to do that as compared to a setting where there's no unmet need. So that's the point I wanted to make.
No,
So I'd like to make a point about the use of off-label data, even if it's not used as a Bayesian in the study. We did some work. We were trying to simulate a study design to optimize it in a rare disease setting, and a study that might be in unapproved areas. And the literature was very sparse in these rare diseases. But when we went into electronic health records looking for off-label use, we were able to find cases of off-label use in electronic health records that enlarged our data set somewhat and enabled us to make somewhat more realistic assumptions when simulating the study for design optimization purposes. So this is not using the off-label data as a prior in the study itself, but simply to give you a more valid simulation. That's another possible use of the off-label data that would be available in pediatrics and elsewhere.
And I think we'll talk a little bit of that too in the non-concurrent control discussion too as well. So I suggest we move on to the next topic. This may tie in as well to non-concurrent control piece question too as well. But let's just say, if we assume that concurrent controls are now the norm and expected in most platform trials, is there anything controversial in any understanding of the definition of what it is a concurrent control? Or do we all have a clear understanding of what this definition really means? Anyone care to comment?
Yes, maybe let me start, because it's not clear black and white. It's not non-concurrent and concurrent controlled, but there are also nuances and some grayish in between. Because in the context of platform trials, it's even not clear that all concurrent control data can be utilized in a specific analysis. So we discussed it before with Seu Pearl, the diver example, and with blinding. And it might be obvious when drugs have different mode of administration, when blinding is not possible overall. But on a sub-study level, sometimes maybe we can only use the matched controls. And also, if we use a two-step informed consent procedure, you might have some problems. And similarly, if the inclusion-exclusion criteria differ slightly between active drugs, then you have an issue. Or alternatively, if some centers only offer a subset because a drug is not available or a protocol amendment has not been approved, then there are situations where you even cannot share the concurrent control data for various issues. So only the control data of patients can be utilized if a patient could have received the drug of interest when evaluating a treatment control comparison. I think this is a key issue we have to respect when we talk about platform trials. And we have to think more about adjusted analysis, where we weigh the control data accordingly to from which subset they come from. I think this is not frequently done.
No, I've been in discussions exactly similar to this. I think in the last year, I must say many times, and it's for those who are sort of naive to the concept, they get very confused very quickly on what the definition really means. So you have to kind of guide them and walk them through. Kert, go ahead.
So I'm just going to say, in a lot of our platform trials now, we do talk about concurrent controls in the protocol, but what we actually measure is we get a metric for every patient that comes in what therapies could they have been randomized to?
Mhm.
And instead of talking about the concurrent or the co-eligible population, we compare to all of the control patients who could have gotten an active drug. And that includes concurrency because it has to be enrolling at the same time. It includes eligibility. It includes site, oddities, as Franz was talking about. But anyway, I just think it's an easier way to keep track of this in the database and in the analyses than it's usually just described. So just an operational issue.
No, I think it's a very good point, and in other areas too as well. Particularly in immunology, now the CAR T has sort of moved or morphed over to from oncology and hematology into the immunology, where you have we're no longer doing single arm trials, and it becomes very hard in this sort of randomized open label setting to be able to, and you have your control arm is basically investigator's choice. So basically, where you go in and say, okay, what are the treatments that they could be given or would and you basically you can say, and that sort of helps you sort of to identify based on their background, like what control groups they are suitable for if you wanted to say look at things in the same design. So I think what you propose makes a lot of sense. Would you like to comment, Olivier?
Yeah, I was thinking as you were say, I was reflecting if what I have in mind is correct or not about what Kert said around just trying to find the word you said around eligibility versus concurrency, right? If it was or not the same thing. Concurrency is more strict, obviously, but then you could argue, I mean, you're a participant under control, and then you're there, and then you could have received all the other treatments that are there at this time. But you will also have participants under control that would have the exact same characteristics as all the ones that are concurrent to the new treatment, so to speak, but they were just not there from a pure time point perspective, right? So they would be somehow eligible, and they would just be eligible to anything else besides the new treatment of interest. So in some ways, those participants, you could have them as non-concurrent control to support your primary analysis, because from a point of view of estimates, actually we'll talk about that later, they would fit because they represent the population of interest, right? It's just that they wouldn't have had the opportunity to receive that new drug that you would have at that moment. They could be eligible for it, but they were just not there from a pure time point perspective. So I see a tiny difference there, but I thought that's very interesting. Thank you.
Yeah, thank you. Anybody else? If not, we will go to the next topic, which will likely generate some exciting discussion. The non-concurrent control topic, which I think is everyone in the platform the trial space is many are very passionate about. We had a webinar through IMI EU Pearl that Martin Pasch and I sort of co-chaired, I think about 2.5 years ago. And amazingly, the space has sort of evolved a lot even since then. But not me. I remember there were very passionate views. There were some that were very bimodal in terms of what could be done in the space. And I commend the FDA draft guidance that we've made considerable progress in the space, and are sort of at least acknowledging that this is out there, and sort of saying, okay, if one wants to do this, what does one have to do to show that this is the right design? So taking all these things into account, the draft guidance recognizes these things, and I think the question is what are points to consider by clinical trialists when developing such designs? And when we think about non-concurrent controls, we also have to remember, obviously, that these may not necessarily be, or may not be in the trial itself. They could be external controls, and so the question is what role should external controls play in development of non-concurrent controls for platform trials? And when I say external, I can say fully external, or we could say some sort of hybrid control. And are there specific use cases where using an external control as non-concurrent control has the greatest value? I believe Karen is first off on this topic.
Okay, sure. Yeah, happy to make a few comments here. So I don't know if my answer will address all of the questions there, but maybe firstly, just to mention, I do think that sort of in all cases, we should be using external controls to some extent, as Bob mentioned, if nothing else, to really help inform the design, really understanding and modeling the available clinical trial data, the real world data, to understand expectations of that control arm, aspects that may impact those rates, and really being able to evaluate consistency of that control rate over time, as well as sort of the between trial variability in that rate. And so I think just, number one, I think they should always be used, if nothing else, in the design stage. And then I think, certainly in cases where they're being considered to be formally used in the analysis and, impacting the design, a lot of the insights gleaned from that type of assessment could help inform whether or not it's appropriate to be using either external controls or non-concurrent controls, and understanding that shift in time. Cases where maybe we touched on it earlier as well, where the endpoint is more objective, maybe there's less of an impact to things like the difference in mode of administration or randomization rate or other factors, in an endpoint of overall survival or progression-free survival, versus maybe in the case I mentioned earlier, where we have a master protocol in pain. Pain is a highly subjective endpoint, and so it has been harder to really figure out how could we use formally these external or non-concurrent controls. So I think again, the objective level of that endpoint could inform that, and then again, maybe just to conclude with the understanding that variability in these historical studies, and how much it shifts over time.
I guess from a general guidance perspective, is one of the points I think I noted, and probably could have been a question, is and I know there's a new FDA guidance, draft guidance coming out, let's say hopefully within the next year, on Bayesian methods. Does something Bayesian there's really not a lot of Bayesian sort of recommendations in the draft guidance, and is that something that we need to sort of be able to teach people on who are not who have fears of the whole Bayesian concept? I know whenever I sort of explain Bayesian to non-statisticians, the first time it can be a little bit rough. Once you get past the first time, they sort of get it and how it's a different way of looking at the problem. Do we need to have more sort of knowledge out there on how to best do this in the platform trial setting?
I mean, I can comment on that, and I think others as well. I do think that is helpful. We've utilized Bayesian methods quite a bit when looking to incorporate these external or non-concurrent controls. Usually those are, of course, in cases where these are not registration trials where we have done this. We are looking at cases to, more in the registration setting. Typically, we would incorporate some sort of dynamic borrowing as part of that, in understanding the places where the borrowing, when one borrows, we get a different decision than when we don't. And what are those spaces? How likely is it to occur? What is really the impact to the decision ultimately? And of course, there would be a lot of sensitivity analyses that would go along with that. In terms of our experience with proposing these types of designs, I do think we've made a lot of progress, where more and more, as these designs are proposed, both internally and doing cross-functionally with a Bayesian approach, and then engaging with regulators on that, we do see that has increased over time, I think, due to a lot of the CID pilot program and things of that nature. But cross-functionally, medical reviewers, I do think there's a lot of work that we need to continue to think about. How do we educate other functions in particular on and getting more comfortable on a Bayesian approach? So agreed, that's a gap we should look to continue to meet.
So I think, Peter, we brought up utility functions a couple of times, and I think this is a very important place to say that non-concurrent controls and Bayesian methods, as Olivier said, there would probably be more openness initially in rare diseases, pediatric diseases, where there's the need is so clear. And one thing I wanted to say about utility functions that's probably applicable to a number of these issues is that it might not be just one utility function. When I was working with Martin Posh, and we had the first author was Thomas Andre. We published a couple of papers, but we developed utility functions from the point of view of the sponsor, from the point of view of public health, from the point of view of an individual patient, and from the point of view of a health authority, and they were all slightly different. And then we have to think about when we make these trade-offs, and we take slight methodologic risks in order to facilitate the availability of drugs. Which utility functions are we going to use? I would argue it probably should be the public health utility function, and perhaps the health authority regulatory function should have the most influence. Utility function should have the most influence.
Okay. Skip.
Yeah, just a comment again from the perspective of pediatrics. I mean, ICH 11 talks about external controls. I mean, there's really two situations I could imagine in pediatrics. One is where you're using those data to establish not so much a formal inferential comparison between a control group and an intervention group, but some threshold that you must exceed or match in your open label single arm study, which would be separate from a more formal inferential approach where you're trying to then draw statistical comparisons between your active, open label active arm versus whatever external control you're using. And I might just point out that part of the challenge in pediatrics is also given the adult prior, when you're developing a product in pediatrics that's been already developed in adults, the community may well not accept a concurrent placebo control. And so you're really forced to be in a situation where you've got to come up with some way of concluding that the data you're getting from your open label single arm study is interpretable. So it's just there's a brief discussion, very high level, but brief discussion of that in the ICH 11A guidance.
And the community should not accept it if there's not a credible standard of care, especially for serious life-threatening diseases.
Well, we can have a long conversation about what communities should or shouldn't accept under any circumstances, but yes, agreed.
And as far as I'm concerned, for me, it's not whether Bayesian frequentist decision theoretic approaches when you think about incorporating non-concurrent control data, because there are a lot of wonderful methods out how to adjust for time trends. I think the key is really what are the assumptions in the background, because you can make good things with both with Bayesian frequency things, but you can also do extremely stupid things. So I think we as physicians have to learn how to communicate what are the assumptions in the background. So there's really a lot of nice papers by Kert. We have also joined papers where we compare both frequentist and Bayesian methods, looking in time machine or simple frequentist methods. And really all methods depend on the assumptions. And you can sometimes translate the results from one to the other approaches. But I think a key step which we have to take all is, if we think of incorporating external data, then there's no way to strictly control the step error rate. Then the question is, do we have to control the step error rate? Because sometimes people try to translate it and start talking about the average step error rate or something else. I think this is not the question. I think if you say, okay, it's worth going for non-concurrent control data, there's no point in pretending that we achieve strict step error control, because that's not the question, especially if we talk about rare disease or pediatrics. And I think one main argument is, we see so many single-arm trials. Why wouldn't it be better to have a small randomized part enriched by external data? I think there we have to go for.
Yeah, it's all about appetite for risk at the end of the day. I mean, it's like what Franz said, right? I mean, there's lots of methods, and I mean, your group, Franz, with Martin, you wrote a very nice paper on the use of non-concurrent controls, and it's all about model specification or misspecification. I mean, if you hit it right, you gain power. Again, power, you maintain type 1 error control, and sure, you have to communicate it right. But people can say you can't guarantee to me that the model is correct, and either they want to take that gamble or they don't. And there are moments where you're on the wrong end of the stick. You know, it's just that. And
if I may just provide a clinical perspective on that, again, out of ICHE 11A, and just I was on that working group that developed that guidance. I agree that the assumptions are important in the context of borrowing data, which effectively is what you're doing, whether you do a frequentist approach or Bayesian approach, you're borrowing data. You cannot have a strict control of type 1 error, and that's pointed out in the guidance. But there's also a sentence that says that the degree of tolerable uncertainty, meaning what is that type 1 error that you're willing to tolerate, is a clinical judgment, not a statistical one, which goes to the fact that, to that point, Franz, of the assumptions that really need to be clarified around that uncertainty that you're willing to tolerate, which is going to vary depending on disease and condition and alternate treatments, et cetera, a very complex set of issues.
Could not agree more, Robert.
Yeah. All right. So I will say that I have somewhat given up on this topic temporarily, but it's more of a stealth giving up. There's so much effort in the world right now going on borrowing historical data into non-platform trials. There are massive efforts on using real-world evidence. It feels, I'm going to say, slightly inconsistent, and I have much stronger things to say, that we're going to end up in a situation where we can use real-world evidence, but we can't use the non-concurrent control that we enrolled yesterday in the same trial. So I'm waiting this out to some degree. I think Franz said something that's really important that I want to come back to, which is saying what the assumptions are. I often hear the issue with non-concurrent controls is changes over time. It's a very specific kind of change over time with regard to changing treatment effects that matter. And so that's part of the assumption. And that's an assumption that you see in meta-analyses. So again, when somebody says, a meta-analysis is the highest level of evidence, but I won't take a non-concurrent control, it's sort of, how does that actually get justified? And then the final thing I'd like to say is, I don't think we should ever be skimping on concurrent controls ever when we can actually use them. The question to me is, non-concurrent controls in a platform, they exist. Do we get a better answer if we use them? When ISBS 2 was running, for example, we weren't trying to avoid concurrent controls. We weren't minimizing, but we've got a decade worth of non-concurrent controls. Do we get a better answer if we include them as opposed to not? As I think Franz brought up, Franz, I'm agreeing with you on everything today. But is it a mean square error?
This must be disappointing for you, Gert.
No, it's great. It's fantastic. Happy to be here.
But how do you know the answer is
discussed in adaptive design for many years. Are you really looking for a design which is unbiased, or shouldn't we look for designs which have the smallest mean squared error?
Yeah.
Skip, you have your hand up. What would you like to talk about? Go ahead, Skip.
I wanted to just draw attention to a comment that was made, a question, and let me reframe it. The difference about the use of non-concurrent control data, meaning it's truly yesterday versus, or a year ago, versus concurrent control data where there's a slight difference in the population. I mean, I think Franz had earlier mentioned some of these issues around randomization. You know, if an ISA has a slightly different eligibility criteria because of safety issues, et cetera, what are the issues in using those concurrent control groups, but where the patient is in fact not quite the same as would have been randomized? That actually, the question, I was beginning to wonder that in myself. Are there ways that we can adjust for that difference in patient population that would allow us to use those concurrent but non-comparable or not entirely exchangeable patients? Thoughts?
There's a Bob, I don't know if you're responding to, there's a question in the chat from Greg Levin that is
right, that's what I'm asking is can people respond to that?
Well, I mean, I think we have to avail ourselves of whatever data there is that would allow us to estimate the influence of the population difference on the treatment effect, right? You know, and if we don't have data to assess that, I think it's hard to know whether the difference in the population or the difference in time is going to be more important.
I mean, you going back to the questions in the chat, you could argue that that's what people do in causal inference, and when they talk about targeted trials, and here we have to learn how to mix these techniques when we analyze clinical trials. But I still have a split opinion. If you have a proper randomized clinical trial, why would you think of using methods coming from causal inference, and why using inverse probability weighting? But I'm not expert in these things, so it's quite a naive statement.
Olivier.
Yeah, and there are also assumptions behind that to say, let's take someone from an RWE database and let's try to make it comparable to an RCT. Let's pretend they were concurrent in some ways. There are assumptions behind that. You're going back to, are people willing to accept them? I mean, the question of Skip to say, what's the cost of including a participant that is not quite exactly to the ones we're randomizing, is that potentially that participant is different. Maybe you can demonstrate to which extent that participant is different. You may want to take that risk, but the person you want to convince maybe doesn't want to take that risk, and that's again the whole problem, right? It's just at the end of the day, to whom belongs the decision to accept that you're submitting a package of which the quality of evidence departs to the one you would have submitted in an RCT. It's just that, you know, as soon as you move from an RCT, you lose all the things that it guarantees you, and there are certain settings in which people don't want to accept that. So it's just that.
Yeah, and we've been talking about a case-by-case analysis, but I think one other thing that and I was in industry for almost 20 years before I joined academics, but I think that sponsors are reluctant in phase 3 to adopt paradigms that are discussed with health authorities on a case-by-case basis, because they're incurring a risk in phase 3 of non-acceptance by the health authority, and so in spite of the fact that most of these things are best considered on a case-by-case basis, any sort of general rules or guidelines that can be developed, and obviously this guideline is a big help that's already there, but the more for sponsors, I think, to apply innovations, anything that can be done to reduce the upfront regulatory risk is going to be result in increased application of the innovation.
Okay.
So,
I think go ahead, Karen.
Oh, I'm sorry. I was just going to say, and I may not be fully addressing Greg's question here, but I would just say I think one thing that matters is the reason that the patient would not be eligible for a given drug. So, for example, if a patient is not eligible due to something that is maybe a concomitant medication use that's not related to the outcome, maybe that's less of an issue than if that patient is not eligible because of some inclusion criteria that affects the severity of the disease and therefore would impact the response and relevance to the other arm. So that would just be one more thing I would add to this question here, is maybe understanding the reason could help.
So just a quick interlude. So like the Viele trial, one of the exclusion criteria is a gold allergy because one of the treatments involves gold. And I don't think anybody expects that affects results.
Yeah.
But
Yep.
anyway.
Okay, so let's move on this to the next topic. Our statistician's favorite topic of estimates, which is not talked about in a lot of detail in the draft guide, but it is a topic that obviously, as we design these trials, is an important topic, particularly if you're trying to establish some level of consistency of these things across substudies. Are there any different points to consider with estimates when defining the attributes in platform trials, and how and when we acknowledge there are going to be some difference at the substudy level, and how will this impact invitation general estimates that can be used across all substudies? Go to Olivier first.
Yeah, thank you. So, like you said, I mean, my first reaction when I read the guideline is that there was no mention about the word estimate at all, and I think several of us reacted to this, and that's even more surprising that when you read the E9 addendum, it's written really clearly that it applies each and every time you want to estimate the effect of an asset, let's say, of a treatment or a vaccine, and then by extension, that applies in a setting where your objectives require to run a complex innovative design like a platform trial. So that's a comment we made actually to FDA. And I have to say, talking about platform trials for a couple of years now, that's a question that I had often asked about how do you frame the estimates for complex innovative designs for platform trials? To which extent is it different? And in some ways, I thought the question was phrased by me, including by me, is not how do you use estimates for complex innovative designs or for platform trials, right? Is your estimates would drive the need for a specific type of design. That would be the right way to think about it. So that was my first reaction, is that yes, that applies here as well. And then very naively, another really simple conclusion is to say, well, you have several treatments, for example, of interest that you would compare to a comparator. Well, that means you have several objectives, and one objective equals one treatment equals one estimate. And then if you decide to add up a treatment, then by extension, you're adding up an estimate. And if you're dropping one, then you're dropping an estimate. And that's basically what we wrote in our paper, among other things, because it deals with basket trials as well. But that was one of the points we discussed. Maybe one point, two key points actually that are interesting from the point of view of estimates for me, and more specifically on platform trials, is that surely when we write the estimate section for say a master protocol encompassing comparisons of several treatments versus a common comparator, is that there would be some commonalities between the attributes of the estimates. So I don't know if you want to project my slides. I mean, I have a table.
Yeah, I can do that. Absolutely.
Slide number 4. I think that will just help a little bit, just to be a bit more visual. But then obviously, when you have an estimate, you have a treatment component. You can go. I think it's 19 on your screen. Yeah, the one just above.
Okay, there we go.
Okay, so we have our five components, right? We have the treatment, the population, the variable, intercurrent events, population level summary, and at the end of the day, some of them, of course, because they're being studied within the same study, you would have some commonality. So the treatment would be different because you have different treatment of interest. That thing would change. the population, in the grand scheme of things, you would expect it to be the same. You could argue, going back to discussion we had at the beginning, that maybe not all participants will be equally randomizable, for lack of a better word, to all the treatments. But give or take, they should be fairly similar. The variable, more often than enough, would be similar as well. But in theory, you could argue different drugs could target different aspects of the drug. Certain could look at remission, other could look at severity, for example, and that could be still done within the same population, within the same study. And then you have the case of intercurrent events. So this is interesting because some of them would be population specific. So for example, change in background medication, but in other, they could be treatment specific. So they would change with the treatments, like so for example, positivity to anti-drug antibodies. And then you would have the population level summary that would also change with the variable. So that's one thing really that sprung to mind when I think about estimates for platform trials. And then the other thing, if you go a little bit further, is how do we deal with the change of control? I think that's a very interesting question. Can you click on the next slide, Peter?
Yeah.
Yeah, so you will need to. So let's imagine you have this case, right? You have a platform trial. You start with 2 drugs, drug 1, drug 2, versus control 1, and then you add up drug number 3 later on. And then as time passes by, can you keep on clicking, please, Peter? Then the control changes. And then what do you do then with the comparison, right? Is that initially drug number 1 and drug number 2, if you keep on clicking, they are meant to be compared to control 1, and you will still have the wealth of data necessary for that. But for drug number 3, it's a little bit harder because initially the intent was to compare it to control 1, but then things have changed, and then it's mainly now concurrent to control 2. And then how do you do with this, right? The logic of estimand, if actually you go to the next slide, is to pre-specify the comparisons of interest. But then the question we need to ask ourselves is, can the original estimand still be estimated? So basically what the slide says is that there are different scenarios. Either you're interested in the comparison versus the best control, and then it means something has happened in your study that forces you to change post-hoc your comparison, and then by extension your estimand, because the comparator is different. The population might change because of the ineligibility to the new standard of care. You might have new intercurrent events. And they will have to decide which comparison is primary now versus control 2, because that's now the new benchmark, or versus control 1, as was intended, even if it's now somehow obsolete. Or another way to look at it could be, well, actually, I'm interested in comparing my treatment against whatever the control is, right? And then if you can anticipate that, you can phrase your estimate in such a way that the comparison will need to be done versus whatever is available at the same time or even before. And then probably you could use both control 1 and control 2 to do that. So that was the 2 points I wanted to share on the topic. But then again, although I love the guideline, my main regret is that there was no mention about the E word in its description. So thank you.
Olivier, I have a question, which is that so I think, so I agree with everything that you said, and the question is, how much of that could be a prospective agreement with the health authority, where you say, we understand that we're in a platform trial, that the controls may change, and then you propose some kind of decision rule that allows you that allows you to decide whether you are going to use control 1 or control 2 or both. How do you decide that? What are the criteria? And you negotiate that prospective rule with the health authority in advance rather than in the heat of the moment. Is that possible?
Well, I think you can try. I mean, now I left the regulatory system. Now I spent much longer time in the industry than in the regulatory system, so probably the thinking has changed. But I suppose the good thing is to try first off, is to trigger the discussion and explain to people what you want to say. I mean, it doesn't harm. I think that's the first thing I would say. Try to be as prescriptive as possible, because that helps you as well with your own with your own thinking, being so clear about what you would do and try to think about it up front is, of course, you're right, is to try to have some form of regulatory buy-in, but it's also good for you once you have the data to avoid reacting, to use your expression, in the heat of the moment. And to be honest, that could apply outside the regulatory setting. I mean, we had a pandemic where we had several large, platform trials, recovery, discovery, where things changed, as time passed by, and it's about the quality of the quantity you estimate. So beyond the potential acceptability by regulators about this strategy, it's about clarity of thought and pre-specifying what we want to estimate. I think it's just good practice. That's what I would say.
Anybody else want to add anything? This was a very good discussion, and an important one, I think, too. So now I'll move on to the next. I think we probably have time for one more topic. Then we'll get some time for audience Q&A. We all recognize that multiplicity is a complex topic in platform trials. So the question becomes, is it realistic to define a testing family within platform trials, even at the cohort subset level? And the answer is yes. Then the question, how would multiplicity be controlled when, say, you have monotherapy and combination therapies in, say, a factorial design that way in the same platform trial? And the concept of platform-wide multiplicity control, which is mentioned in draft guidance, is this really achievable? I believe Karen and Olivier have thoughts on this. So let's just say who wants to go first.
Karen, did you want to go first already?
Yeah, I'm happy either way. I actually was just going to sort of pick on that last part of that question in terms of platform-wide multiplicity control. I think somebody already answered this, indicating this is probably not achievable. I also just think it's maybe something I don't know why we would want to try to do that. So I think there are many reasons why that's maybe not advisable. I do remember many people have talked about this, Liz Levange in particular, regularly reminding people that, if we try to do this, especially for these external master protocols, it would be very hard to get companies to agree to submit their molecules into these, and would prefer to probably just run the trial internally. So again, I was just going to kind of comment on that last part there of hopefully it's not something we're seeking to do. It would be very, very hard for one thing, but also I think would really impact the ability of the platform trial to progress and serve patients.
Thank you, Karen. Olivier.
Thank you. Yeah, I mean, Karen, you stole my punchline. That's exactly what I was about to say. No, I was going to say it's not so much about it achievable. Is it more a question? Is it advisable, right? And I would have quoted Lisa as well. I think that's the key question. I mean, yes, it's achievable if you want to use very small alphas, but who would want to do that? So, like I was saying in preamble, I mean, just to put an emphasis on that, the very big win for me in the guideline was really to say, again, if your different treatments, they support different regulatory summations, that's okay. Go away with 5% decided per treatment. I think that was a great win. Other than that, if you start looking things that are different variants of the same treatment, and each of them could give you an approval that you probably need to adjust for the alpha, I think that's fair. I mean, there are probably 20 to 30 papers discussing that. I was very happy to see that now cast in stone. The thing that made me a little bit more, so that's about family-wise, you're right, to be precise, assuming we can define what's a family in that case. The thing that made me at times a little bit more uncomfortable, it's more about the other types of error, and namely the risk of multiple false false decisions. Actually, maybe if you can bring my slide.
Yeah, I'll get you slides. Yeah,
ahead.
one second.
Yeah,
Yeah.
and I have one that explains that very nicely as well. I'll tell you which one that is.
I can go there.
Yes, yeah. If you scroll
That's the
down,
ones at the end. Yeah, I got it. Yeah.
yeah. If you scroll down
To
again,
here.
yeah, tell you what it is.
Is it this
No,
one? Next one.
down again,
This
again. No,
one.
you have it's a bar graph. Yeah, you'll see this bar graph with colors.
Okay, this one. Okay.
Down again,
Okay.
it's a bar chart.
Okay, let me see. Okay, see. This one, gotcha.
Well,
Okay.
yeah, exactly. Yeah, thank you. So that's just to illustrate my point. It's going to be well more visual. I mean, one of the pushback we received at the time discussing the control of type 1 error, particularly when I was at EMA, was of the risk of multiple false decision. And then one way to understand that is via this graph, right? Is imagine that you have several experimental treatments. They all have a certain response, like the bars from red to yellow, and then the control arm, as observed, is the one in green. And then if you just press one more time, please, Peter.
Yeah.
But then the truth of the control arm is like that. Then of course you can see the number of type 1 errors you're making. And then the point people were making was to say, doing at the time was saying, well, if you were to test all those treatments in separated studies, then you would have a random law maybe only once, right? Whereas here you have a random law that's going to end up penalizing all your comparisons. You're going to declare multiple treatments as being false positive. And I thought, when that point was raised to me, I thought that was a very fair point, and I thought that was understandable that people wanted to push back on it, from a pure philosophical point of view. But my counter argument to that was to say, first, you would have to know it's a random law, right? You don't always know that. You will need to be able to provide the meta-analysis to characterize a little bit the response of the control arm to see whether or not you had a random law. I think that's one aspect. Being able to prove that it's a random law, I think that's important. The second aspect is that having a random law doesn't necessarily mean that what you observe is a false positive. Is that it induces several things that are being positive and they might be wrong in hindsight, but we don't know that, right? So that was the other aspect to it. And then finally, an argument that was made at the time is people would say, well, a logic behind the family was all right is to say, well, if you run into a platform trial, we could ask you to control your family wise all right to a similar extent to the one you would have had in separate studies, which I think is a point that can be defended. But if you wanted to do this here for the risk of multiple false positive decision, the local alpha you would need to use, it's actually a very nice paper from like 2018, I think, from Howard. And that explains that very well, is that the local alpha you will need to use for all those comparisons is even smaller than what you would use for Bonferroni correction. So you end up killing all your chances to making any form of statistically significant claims. And then, of course, by extension, you would control the family errors rate, but we already said we don't care about it anymore. So I thought philosophically I could see the argument, but I also thought that practically, could we really prove there is a problem? And then second, addressing it, I think, would again defeat the purpose of having a platform trial. So that's the point I wanted to bring on multiple things. So thank you.
Thank you. Very well
So
said.
I just want to make two comments, Peter. One is that as Olivier and Karen both pointed out, you're going to end up foregoing the benefit of some of these innovations if you insist on family-wise type 1 error control across them. And I think that's an important point, and it brings us back to the utility functions. I personally think in most cases that you have to think about what individual trials that the platform trial or other master protocol is replacing, and what would be the total type 1 error rate of all those trials. And so that would be a more permissive, but I also think justified. And obviously, Olivier's remarks touched on that as well. You also have to consider if there are cases that are intermediate between the two cases that Olivier raised of complete independence and obvious dependence. If they're intermediate cases, then I think you have to think about things like the unmet medical need and so forth that have been raised before, because we don't want to be so rigorous that we end up not developing drugs for the cases of highest unmet medical need and rare diseases where you can't get a large sample size and all these other things.
Very good. Anybody else?
So I would just one quick thing. So I wouldn't adjust unless you're going to adjust the separate trials, the platforms replacing, just as a general rule.
Yep.
Cindy asked about common mechanism of actions. That would be a situation where you might, but you'd adjust for I don't think you would run multiple separate trials. So I think that's fair. One thing that's missing from the multiple type 1 error is just what is good in that sense. You have a higher chance of multiple type 1 errors. You also have a higher chance of 0 type 1 errors. So, how do these get weighed before we decide what statistical thing we ought to do? We ought to decide what is the metric that we're trying to optimize in the first place. And I'm not sure emulating separate trials is obviously the best. We should decide what's best for society and maximize that. And I think in many cases, getting 0 is very advantageous and would be viewed as a plus. Nothing is being used off-label, designing new trials in the future. The control arm is still pure to some extent. Anyway, I think we should think about what the metric is before we decide to maximize nothing.
I think that's a fair point. But at the end of the day, I think it's the heart of the discussion, like as always, is that can you convince someone who's using a certain metric that he should let you use that you would want to use a certain design based on another metric that you think is more valuable. I completely do take your point, but if you come back to regulators, for example, because at the end that's what matters from the point of view of type 1 error control, and you say, I'm not sure I'm controlling type 1 error, I'm not sure I'm controlling the risk of multiple false decision, but my utility function is completely clean. They can say great, but the mandate they impose on us is not based on an utility function. They think based on type 1 error for the best or for the worst, right? And unless there is not a change of mentality around that, I'm not necessarily saying that they should or they shouldn't be one, but just for the sake of the argument, then you're down to entering a discussion with people who use a certain metric to make decisions. It's just that my issue.
So I guess I feel like the purpose of a discussion like this, should be to try to stimulate evolution of the default thought processes of health authorities as well. Congratulate for the great guidance. This is, to me, this is a very important point. Is that rather than thinking in terms of type 1 error, I would like to see health authorities evolve towards primarily thinking in terms of maximizing public health. So I understand that might, and they obviously they do think about that, and they make very well considered studies of individual cases where they make exceptions based on that. And I think it's okay for us to encourage that and propose things that might not be standard today and might not be easily agreed to.
While we're on that topic, I would like to be able to at least take a couple questions from the chat. While we're still on this topic, there's one related multiplicity. Does anyone want to sort of talk about the situations? I know this has come up in a lot of my own sort of consultations with the agencies. How we sort of want to deal with situations where there are different drugs coming from a similar mechanism of action. I know this will be a topic within the factorial design, as well as when we do multiple combinations with the same backbone. And we know the backbone perhaps drives the main efficacy, and another component is just an add-on. Do we need to adjust for multiplicity in this scenario? I know I have my own views. I'd be interested in the views of the panelists. And may want to take a stab.
I will take one tab. What worries me in these situations is that you basically encourage artificiality. I mean, if I'm going to explore 8 drugs over 4 years and you start doing this, it's, maybe I can do 3 at a time. You start encouraging people to go, well, I'll do 3 and then I'll say that it's a new trial. And essentially I'm artificially avoiding multiplicity penalties without actually generating a benefit for society. And so I definitely want to avoid that. Now there's to your question as well, but.
And I think for drugs from a similar MOA, that's exactly the problem. Whereas multiple combinations from the same backbone, if they're really different MOAs, those would have had to have been investigated separately. They're separate hypotheses.
Good. One other question that does come up in the chat as I go, we answered Dr. Levin's question. Dr. Yako asked the question earlier on. His question is, what is the definition of blinding? Is it that the patient doesn't know which of the active treatments they've been allocated to, or is it that they don't know whether they've been allocated to one of the active treatments on one side or a placebo on the other side? Or is it they know that none of the available active treatments have had the randomization probability more than, say, 50%? There are obviously different views, and many other differences could be suggested. Highlighting Franz's suggestion of full blinding suggests that we may need to work with different definitions and discuss which apply to a particular trial. Who has some thoughts?
I think this is a really case-by-case setting. It also depends on how you're doing randomization, 1-tier versus 2-tier. There are other forms of partial unblinding. People have side effects, practical issues. Everybody gets an oral, but the pills look a little different. Some people may be savvy enough to look that up and see, some may not. So anyway, I think this is really a case-by-case on what do you think patients know and how do you think they change do you think the treatment effect is changed on the basis of that knowledge?
Yeah. Anybody else with some thoughts? Are there any other questions from the audience? I know it's Friday evening, and it's in Europe, so people are tired. So, there were a couple questions we didn't get to. We might be able to. Let me sort of see here.
There's a new one from Ronan Fitzpatrick.
Okay.
So I'll take this one. It's on Bayesian. I think on Bayesian. I certainly think one of the great things here is just the extra experience that has come out of Bayesian trials in terms of borrowing information, in terms of exploring all these variance bias trade-offs and so on. I think all of these issues have been explored tremendously more in the last, especially 4 years, than they would have been without platform trials. Surely that will inform Bayesian guidance, and it's good to have real world experience and see what actually happens when you put it into practice.
And I think it also helped on which supportive analysis are needed to show how robust the results are. I think this has really increased a lot by looking into platform trials because you can reveal the issues.
Great. Anybody else want to share any last thoughts?
I guess one last thought I want to say is it was very interesting. Everybody here came from different perspectives, and we had meetings prior to this. And at the end of the day, I think we agree on 95% of what's there. And of course, our disagreements are interesting, but it was good to see. It was good to see you always sit in your own office and go, oh boy, I hope I'm not the only person wrong about this in the world. But it was good to see all the agreement.
We agree. And I want to thank everyone for their participation today. And it's been a good discussion. And it's just sort of a chapter in the journey, right? As we continue to as the external environment keeps becoming more and more complex, and we're sort of pushed to be able to bring new innovative therapies to patients who need them. We would have to use our creative minds to come up with the best design that's going to meet our situation, so that we can deliver drugs to patients in need. So I want to thank everyone. And the slides for those attending, these slides will be made available and the recording will be available probably hopefully within the next week from the meeting, so people can look at it at a later point. If there are any questions, please don't hesitate to reach out to any of the speakers. I want to thank everyone and wish everyone a good rest of their day and a wonderful weekend.
Thank
Thank you, everybody.
Thank you, Peter.
Thank you.
Thank you,
Thanks a
everybody.
lot. Thanks, Peter.
Bye now.